I thought I should do a recording of the Quinoa presentation that I did at CSV Conf earlier this year. That was held in Puebla, Mexico. So I had the slides in Spanish and then I spoke in English. I've reworked the slides now to be in English, but in the description I'll link to both the English and Spanish language slides. So I did a little about me. I know a little bit of Spanish from working with Plan Cebal in Uruguay. Later, I've worked at a bunch of different web development projects, including Museum of Modern Art on their website. More recently, working on open source mapping software with gerrymandering, anti-gerrymandering projects. Started getting into language models in 2020. I gave an example of a sequence-to-sequence model in Spanish because that would change the grammatical gender, and sort of got involved in these different Spanish language model materials. And I am still looking for a new job. There are three parts to the presentation: talking about the biology, getting everyone up to speed on that, recent large language models in this biology space, and then evaluating my own projects. Big wall of text, all this DNA data. We live in a very amazing time where, in at least my lifetime, we've gone from knowing very little about what's going on in cells to having the full genome of several different plants, animals, and humans online. You can even order your own DNA to be sequenced. But then you download this gigabyte or gigabytes of letters and you realize, oh, wait, what am I supposed to do with this? I'm not a scientist. I don't have a lab that I can do something with this. So let's find a different way of viewing this. So this is a genome browser tool. This one's created for rice. And essentially you can zoom in at different levels until you're seeing parts of the DNA sequence that have been labeled or annotated. In this case, it's showing some proteins that have names and some kind of description for what they are, what they do. And then there's some that we know they're a protein, but they don't have a label on them of what they do. And within those proteins, there's every three DNA letters make up a codon, and that codes for an amino acid. So a protein sequence would start with a start codon, codons representing those amino acids, and then a stop codon at the end. It can go in either direction, and there's still lots of gaps in between these proteins, which is the non-coding DNA, sometimes called the junk DNA. But there's a lot of features to this in terms of how our cells figure out where they are in the organism, or how often they copy and express different genes. So there isn't like, these are the good parts, these are the bad parts. It's more like, here's one way of looking at it. Within this system, there's all sorts of oddities. There's palindrome DNA in your own biology, and there's also overlap or frame shift. I was saying before, every three nucleotides would equal one codon, but there's some that are overlapping with each other depending on where they read, and they're connected to each other and very important. So there's a lot of work trying to understand how this affects how they're inherited and affect the body when they are so closely connected genes like this. I've tried to divide things that people had heard of in biology into three different categories. The first being these well-known, well-understood things. So probably something you would have learned in high school would be you can have a mutation where one of your DNA letters, instead of being the majority or norm for that letter, is something else. And that affects the amino acid, and then it affects the protein involved. So SNP is a single nucleotide permutation, or one letter is changed, and pretty straightforward. You have it, then something happens. You don't have it, then you don't have that outcome. So those are pretty easy to understand. The next category of example that I gave was things that are innovative, things that people are studying a lot right now. example I gave was polygenic scores. So I uploaded my DNA to a service called nebula.org, and for something like sleep duration, which is a health trait, there isn't like one switch there where it's like some people have this and some people don't have this. Instead, they've looked at 84 different points in the genome and assigned different linear weights to them. And then they're calculating at the end that I'm in the second percentile. I'm not doing very well in sleep duration here. Now, there are a couple things I didn't get to say in my talk that are relevant to this. One of them is it's so important to know that if there are these dozens of different places in your DNA that affect this, it's not realistic to say that we know what each one of them does, or that I'm going to start flipping switches and it's going to change my sleep duration. It's more to say that end health goals in the real world are very complex based on this. The second thing being that um, this doesn't affect, if I want to get better at sleep duration, I shouldn't be waiting for, a gene treatment, gene therapy. Really, what I could do is have, a better bedtime routine, avoid things that are loud and wake me up in the middle of the night or something, or, different, there's so many things you could do that have nothing to do with genomes. But this is a good example of something that is related to your genes kind of as a baseline. And then I wrote pangenomics, kind of a similar idea that now we have the genomes of so many people in a population, we can start to make these kind of broader traits and understandings of which genes are relevant to what particular functions. And then I think a lot of people have heard of protein folding, AlphaFold models doing a lot there, and then CRISPR-Cas, which is a combination of techniques that existed in the natural world and are now being harnessed to cut, copy, and paste DNA. Final category I said is speculation. Now, epigenetics, gene drives, these are all real things that exist and have research on them. But, if your friend listened to a podcast and he's like, you gotta do this epigenetics thing, it's so important. We're not there yet. Don't just listen to anybody with their different theories that are on it right now. I think there's a lot more that we don't understand about these things. I also want to make the point that people think that, now you're going to design genes and cells that do different tasks or have a different abilities, like the X-Men or something. But it's more realistic that, like with CRISPR, what most people are doing is they found genes that they want to cut, copy, and paste into other organisms or remove from somebody. And those are traits that already exist in the natural world and are just being like turned on or off. So this talk is about quinoa, which is an unusual choice, but I once gave a talk about quantum computers at a nuclear conference, and it was very out of place. I didn't think any of those people were going to give me a job or anything. But the goal of that was I believe in some kind of international peace discussion, conversation, some kind of technology to help people helping each other out and sharing information about technology. And then at the end of the day, I say, well, you believe in this, but what are you doing about it? And I think it would be a very serious issue if, if I'm saying that there's a problem with the global food supply, climate change, things like that. So what am I doing about it? Maybe I should learn something about it. Maybe I should find like some space where I don't know a lot about it, but it seems important. So that's why I'm going to talk about quinoa. It's a pseudo-cereal grain that grows in Peru and Bolivia, mostly. It's related to a few different plants, including Quinoa, which is also in Peru and Bolivia, and Huazontles, which is primarily in Puebla, Mexico at this point. Um, that was where the conference was. I went to a restaurant and had Huazontles with pipián, which is like a pumpkin mole sauce. It's very pumpkiny. And then the huazantles themselves are kind of a green, soft, leafy, grainy quinoa thing, which they had rolled up and boiled to be soft. And so you're eating that. A lot of fiber, but now I know what that is. Now I've had that. And then quinoa is something that's a bit more common and you can buy on Amazon. Um, Now, quinoa, interestingly enough, and this is also true of horizontalis, now that they've done some research more recently, they're a hybrid of two extinct plants. And sometimes people read that and think, oh, they're the two parents, and then they combine sort of like a mixed plant. But actually, it's polyploid, meaning the full genome of both species combined into the cell. And it has both of those subgenomes still today. Lots of different crossovers can happen. If you think back to high school biology and like how plants and animals work, that seems pretty bizarre that for each chromosome you would have four copies instead of just two. But this happens a lot in the history of plants that we eat. Something I didn't know about until I did this research, but it's something that affects the grain size, and there's actually been like 100 years of research in trying to get this to happen um, sustainably in rice to make the rice more profitable and plentiful. Anyway, there's all these research centers around the world that are working on different crops, and they're all linked to each other. And there's also different universities and national level centers. But when it comes to like what's the global leading center, corn and wheat are researched in Mexico. This is where the Green Revolution strains of corn and wheat came from. Potato is researched in Lima, Peru, where a lot of potato varieties originated before the Columbian Exchange. There's still a huge percentage of potato varieties in the world still are only grown in the Peru region. I think they also do some research on quinoa since it's also more local to them. And then FLAR is a rice growing institute for Latin America. The sort of more global one is in the Philippines, but they have different outlets around the world that are working on this. So why quinoa? I wanted to make a point of saying that we can learn a lot from traditional agriculture. People have been growing quinoa at all sorts of elevations and all sorts of conditions for thousands of years. So if you were going to grow quinoa in your backyard, you wouldn't just kind of do it from scratch. You would want to know whether someone's done that before, what kind of seed you would use, that sort of thing. Uh, People are interested in quinoa as a crop that is able to handle salty soil, heat, drought, high altitude, low altitude. Particularly, it's useful for climate change. I didn't get to talk about this so much, but all these research centers that are working on agriculture are very invested in finding projects they can do to talk about their relevance to existing technologies and problems. So, if you're doing research on rice in Southeast Asia, you want to talk about climate change. What happens if all the millions of people who depend on that rice crop start having problems of some type with their rice? And the kind of the go-to in these different environments, these different research centers, is to say, let's do a trial of how would quinoa work as a replacement. So there's quinoa research on the Mekong River Delta, there's quinoa research in Japan, in China, in the UAE and Saudi Arabia. If you look at research papers about quinoa genomics, you will be surprised who is showing up in these research paper centers and things. Now, the research of crop cultivation and genetics follows the original traditional research. So, for corn research, there's all these different varieties people were growing already based on different preferences. And so they'd say, oh, this is a wheat that works this particular way. What's the gene that explains that? Whereas with quinoa, there was fewer agricultural research to build on. It's only this year that there's papers appearing talking about even very obvious phenotype traits like white, red, and black quinoa. So there's a paper there. Probably the quickest way to bootstrap this research would be to look at the genome of quinoa and find similar genes in other plants. And then, if it's different, try to explain that difference and triangulate it to other plants. And then there's hundreds of other genes that are unique to quinoa that maybe could be predicted based on this. There's also sort of a question of, are you going to model the individual genes, the proteins, or are you going to take the full DNA sequence and work from there? Like, what modality of research are you going to do? I'd also say there's traditional, I hate to say traditional, but there's sort of an existing expertise in bioinformatics that's out there and open source. So there's BioPandas. There's a group at St. Jude's Hospital that does interesting genomics in Rust programming language. So they're out there, they're on GitHub. Specifically for this talk, I wanted to talk about this new development in large language models. So if you go back, there's a protein BERT specifically trained on proteins. Within the last year, there was interest in models trained on the human genome or specific organs within the human genome. AgroNT is the most relevant model because it was just trained on plants, including the quinoa full genome. That's a DNA level large language model. And then early 2024, there's a EVO, which was trained on prokaryotes or bacteria type cells. They removed all of the genomes known to cause human illnesses, and that's from the same team as Hyena, so it's sort of like continued evolution. And then Caduceus, kind of a lesser known model at this point, but they're using the new Mamba architecture instead of a transformer. Now, on the right column, I talk about tokenization. So, for very large, long sequences of DNA, this is really challenging for language models because they have a limited context. And so the AgroNT model would had a token for every 6 nucleotides, sort of like 2 codons would fit into one token. And it had a context window of 1,000 tokens. So all of the tasks and research and evaluations in the AgroNT paper look at up to 6,000 nucleotides, because that's how much they could tokenize and keep in their context. By the time the EVO paper had come out, there were all these things people are doing with Mamba or with the rope scaling, which is the rotational encoding. And so they're claiming to have a 131,000 token context. And so they've decided to do individual nucleotides for that. So that's a huge difference going from 6,000 nucleotide context to 131,000 context. It's also a larger 7 billion parameter model compared to 1 billion. So that's a lot of change just from September of last year to March of this year. And that's when the papers come out, so these are behind, what someone has in a lab already. Some recent evaluations, I mentioned the AgroNT paper. They're doing some conventional bioinformatics tasks, looking at a DNA sequence and then asking it to classify it based on long non-coding RNA, gene expression, promoters, and up to 6,000 nucleotide base pairs. And you can load it using Transformers Auto Model for sequence classification and make these classifiers. For EVO, it's more of a GPT style model. Looks like what they were doing in their studies was measuring the perplexity of different sequences as a way to build a classification system. So you can't load it into a conventional classifier right now. You sort of need to bootstrap it with that code or something similar. For my own projects, one idea was a protein embedding model. So amino acids, there's I want to say 22 or 21 essential amino acids that are in protein sequences, and then there's the 4 nucleotides. So they realized, oh, this is 26 letters, we'll use the letters of the alphabet. So this is a simple sample sequence, part of a protein using that alphabetic code. And then I took some existing protein models, ran lots of different samples and pairs in it, and then retrained a model with a different technique based on those cosine similarity distances. So this chart here is just showing they have pretty similar instincts when it comes to the test set of how far apart proteins should be. There's a evaluation called TAPE, which is looking at fluorescence and other systems where the question is, could a classifier detect a small change? And it remains to be seen, but the protein embeddings themselves seem to be located in a useful way. The other idea I had was pre-training on protein sequences, those amino acid sequences, and then fine-tune on a description. And I'd like to do this so that I could have multiple tasks solved by one model, maybe do question answers with a natural language, or when you have the output answer, you could use kind of existing explainability tools to find out which part of the sequence is it looking at when it outputs the correct answer or the incorrect answer. Um, this hasn't really been working well. I really think this would be interesting to have a mix of natural language and biological language. On the right column here, I have an example of a sequence, the kind of like gold, like original description that I might have for that protein, and then the description that it created. So it gets the vibe, it gets the theme of what kind of topics I'm talking about and want to generate text about, but the answers that it gave are completely different from the original text. So I've seen this as mostly a failure. Um, I'll get back to it. The other idea I had was training on the DNA sequences and snippets of DNA sequences, um, but adding these additional tokens. So for every DNA nucleotide A, C, G, and T, I've added sort of an additional character there so that it won't be triggered by anything else. And then the LoRa adapter will have these new tokens, embeddings, fit into place on top of the model, and sort of combining two different models. This hasn't really been good at when evaluated on the tasks from AgroNT. I'm a little curious whether I could have done the LoRa parameters differently, or if it's there's different things blocking it from uh, solving the task correctly. Like if there's some overfitting, like it's doing it correctly based on whether the training data looks similar to the test data, and then doing it incorrectly when something's changed. I would have to look into this more. Future ideas, um, let me go back to that one. Future ideas, I could see doing this where either I start with the protein model that's working and then add natural language English system, or something that's more recently here is you could merge models. So maybe a working DNA model and working text model, merge them together or use embeddings to sort of let them work to their expertise. The other idea I had is using the existing open source genome browser. Like I showed the example for rice to look at the rice genome. It'd be interesting to set up a site like that for quinoa researchers to collaborate and look at particular DNA sequences. I sort of have a project there where I have a Netlify static site somewhere where people can do that. Um, well, it's just there. You can browse it. Um, the other idea is collaborating on data sets. So the stuff that I used for the tiny llama model is a data set called green bean proteins that I created or collected from Uniprot. And so that's online somewhere, just proteins and their descriptions. And I also modified it so it has just a binary classifier version. If you just want to say, can I train a binary classifier on this? I think it could be expanded to all sorts of questions and answers. In terms of, suppose this is only capable for very large research labs to do these combined natural language and genomics models, or let's say those are coming out in the future and I can't really compete with them. If that's the case, you could still do a lot with what I'm calling winfrastructure. So coming up with tasks that if someone presented you with one of these models, you would be able to test it and find out how useful is this. Example might be, since there's all these existing bioinformatics tools, evaluation where it uses the correct tool to solve problems based on given sequences. could identify, this is a research thing that a lot of the plant genomics researchers are working on now, is identifying useful genes that are in wild relatives of crop plants. Like, oh, maybe this thing from the wild is something that would help this plant grow quicker or be more resistant or something like that. The other idea is identifying genes that help the plants resist diseases, which happen. Plants can catch like a fungus or something. Pests, which are predators of plants, different insects and things. And drought, which is nature. And the idea of looking for them in genomics is good because instead of using, some kind of chemical someone's found and synthesizing it and manufacturing it, maybe you could find something that a plant is using somewhere in the world already that is non-toxic to humans and have the plant produce that naturally. You sort of cut, copy, paste. It's protecting itself from these threats. So wrap up at the end. When you download your DNA, you're getting some big data problems. It's too much for their language model to read a whole DNA sequence and understand it. Quinoa is special in that it's polyploidal. I think I knew very little about how different plants worked in terms of plant genomics, and this turns out to be pretty relevant to how complicated it is as a genome and as a crop, and how variable it is, in terms of different possible crosses you could do with quinoa and its relatives. We're starting to understand a little bit about quinoa genomics, but it's behind other plants and behind because of it not being the cash crop that's gotten all this attention of these other major crops. Language models and embeddings already exist for DNA and proteins. Maybe that's something you could, if you're interested in this, you could kind of find those and piggyback on top of that. These newer language models that have come out this year now have a context of tens of thousands of tokens using rope scaling or the Mamba architecture. This really is a game changer that gets understated because before, like the AgroNT generation of models from less than a year ago, we were saying there's limits to what you can do with this model because it only can look at 6,000 base pairs at a time. Now you're looking at a context of tens of thousands, 100,000 base pairs. The kind of tasks that you can solve with that are maybe it looks like the Stanford team wants to generate their whole genome of a new cell with a language model. Kind of skeptical what that means, but the thought that they could do that now it's now possible because of these larger context models that no one's really gotten a chance to tool around with before. The other thing that I want to leave with is that research and benchmarks that you do, even if it seems like kind of small potatoes, they direct these future models to meet those benchmarks and prove that they can solve these problems. And so that's why I think it is relevant that you could go back and create these kind of tool use and gene identification tasks. And if you even if you don't solve them, future gene models could be directed to solve those. Then I have resource pages. These aren't just like, oh, I wanted to cite something. This is a bilingual CRISPR page, different pages and videos about the biological language models that I mentioned earlier. International research centers, the video about growing quinoa in the Mekong, other research papers. All Fed is more of a effective altruism thing, but they look at food after kind of global disasters, what kind of foods might we be able to grow and recover. And then there's some GitHub links here for bioinformatics and CRISPR technology stuff. All right, muchas gracias a todos. You can follow me on Hugging Face. I want to end this presentation with a recipe, and I ended up with two. One of them is this Tex-Mex quinoa, pretty easy to make. in one pan, and then Rice Krispie treats. Somehow I never thought of this before, but if you get the sweetened quinoa cereal, throw in some marshmallow, you can make a nice Rice Krispie brick with puffed quinoa. So maybe you'll check it out. Maybe you'll make the food. Maybe you'll make language models. But yeah, that's what I have.